We'll be talking in English.
Yes.
So the important links between citric acid cycle and other cycles of the human body are four intermediates can go outside. The first one is citrate. It can go into the cytoplasm when there is excess of glucose in the body, so it is converted into fatty acid. The second is succinyl-CoA, which is also in the citric acid cycle. It is used for heme synthesis. Together with glycine, is the first step of heme production. The third is oxaloacetic acid from pyruvate into gluconeogenesis. And the fourth one is gluconeogenesis. The millet shuttle. Millet was also produced.
Millet shuttle, millet
Millet was
shuttle.
also produced. This one. It can go to gluconeogenesis. Succinyl-CoA can go into heme synthesis. Okay.
Okay.
Like citrate
And
can go
millet
into fatty acid
can go into
gluconeogenesis.
gluconeogenesis, and citrate also go into fatty acid synthesis when there is glucose
Glucose is more than required.
normal. Okay.
So, electron transport chain and oxidative phosphorylation. You already know that. What is electron transport chain? It is basically oxidation and phosphorylation. So, why is it oxidation? I already told you that oxidation is a process in which oxidation number of a compound is increased. So, mitochondrial inner membrane. Both of the process occur in mitochondria. Citric acid cycle and UTC4, and glycolysis always happen in cytoplasm. Glycolysis is universal. Every organism in the world, even a bacteria which have no mitochondria or other organelles, they can also perform glycolysis to produce ATP. It is universal to every cell in the world. So it is in the cytoplasm. Now, electron transport chain function is to oxidize NADH and FADH to generate electrical energy by passing electrons through the electron transport chain to oxygen, ultimately to oxygen. The final step is the electrons which are being transferred from the first complex to the fourth complex. Ultimately, that electron is transferred to this oxygen. If there is no oxygen, ETC will not be working because the final step require oxygen. Okay, and it creates potential proton gradient across the inner membrane. I told you that this moment of electron create a pole inside the cell of proton, which when come back to the cell, it lead to phosphorylation of ADP to make ATP. Okay, this is the force which drive phosphorylation of ADP to ATP. Now, some inhibitors. We have some inhibitor which intervene in this pathway of electron transport chain, and we also have some uncouplers. Uncouplers are those which do not hold this pathway, but they disrupt the pathway in a way that it can produce heat, but it cannot produce energy. Uncouplers are those organisms, those substances. So inhibitors are cyanide and carbon monoxide, which inhibit complex 4. Barbiturate and rotinol complex 1, oligomycin, F0 component of F0 F1 ATP synthase.
Yes.
That's tube we discussed. So uncouplers are 2,4-dinitrophenol, aspirin in high doses, and thermogenin. It is a protein, thermogenin, called brown fat. It is present in neonates and also in some animals, which is required for them to produce energy to keep their bodies warm. So yeah, that's it.
Yes, sir.
Yeah, it's